lactose has to be broken down into galactose and glucose. Which enzyme is needed here? Beta-galactosidase. Frederick Sanger was the first person to develop an automated sequencing machine which will display the sequence of that particular strand of DNA on the computer screen. If it goes and binds to the repressor, it will inactivate the repressor. Therefore, the repressor will not go and bind to the operator region. Hello everyone, a warm welcome to today's session on chapter 6 of 2nd PUC Biology, that is Molecular Basis of Inheritance. I am Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the mechanism of translation, and before that we had understood about the genetic code, the salient features of the genetic code, learnt about the tRNA, which is an adapter molecule, and so on. In today's session, let us study about the regulation of gene expression, and we shall study about a project which is called as the Human Genome Project. So to begin with, let us start with learning about the regulation of gene expression. How does the genes get expressed? So in transcription, translation mechanism and all that, we came to know that the genes are in the form of triplet. They produce the amino acids. Those amino acids are translated to proteins and all that, right? Now, which particular gene gets expressed in which form, we shall look into. So in eukaryotes, the regulation of gene can be studied under the following subheadings, that is at the transcriptional level. So at the transcriptional level, what get forms? The primary transcript, right? So what is the primary transcript there? That is nothing but the mRNA. So first step in the regulation of gene expression is the formation of the mRNA strand. That is nothing but it is a transcriptional level. And next is the processing level. Why a processing level is involved? Because I am talking about gene expression or the regulation of gene expression in eukaryotes. If it was regulation of gene expression in prokaryotes, then this level was not needed. Why? Because now you have to tell me, you have to guess why. Because we had learned that is in the prokaryotes there is only exons present, right? Only coding genes present. The non-coding genes were absent there. So therefore this processing level was not needed. But in the case of eukaryotes, we had learned that, if you have forgotten, please go back to the previous sessions and have a look. We had learned that mRNA strand consists of the exon, which are the coding sequences, and the introns, which are the non-coding sequences. And the introns had to be removed. So if the introns had to be removed, then a particular level has to be undertaken by the mRNA molecule or the mRNA strand, which is called as the processing level, wherein the splicing mechanism will occur. And we had learned in splicing how capping will occur, how the methionine will go and bind there. All that we have studied in the previous session. So therefore, processing level is also, we have learnt about it. Next is transport of mRNA after the transcription process. That mRNA, so this transcription process and all that will take place inside the nucleus of the cell. Say for example, if this is a new cell and there is a nucleus. So this is a nucleus. So all the transcription process that is there will take place inside the nucleus of the cell. Now what happened in the transcription? mRNA got produced, right? mRNA got produced, then the processing of the mRNA took place if it was a eukaryote, all that. Now this mRNA strand that is there inside the nucleus, that has to come out into the cytoplasm. It has to come out into cytoplasm. So the mRNA strand will come out into the cytoplasm. Once the transport of mRNA from nucleus to cytoplasm occurs, then the translation will occur. So in the previous sessions, we had studied about transcription, translation. Now where is the mRNA strand? In the cytoplasm. Once it comes from the nucleus into the cytoplasm, the translation process will occur. So that is how we study the gene expression in the following steps in the case of eukaryotes. If it is prokaryotes, processing level and all that is not needed. So we'll study in detail in eukaryotes, what exactly happens, how the genes get expressed by taking certain examples. So the genes in a cell, in the case of eukaryotic cell, are expressed to perform a particular function or a set of function. So we all know that the genes, they code for a particular amino acid. Those amino acids get translated to a particular protein. That protein is nothing but an enzyme, right? Because proteins are nothing but enzymes. So those enzymes will actually participate in the certain function, a specific function it will help in. Say for example, our hair is made up of keratin. From where will keratin come? Always they say that you need to have good things and you need to oil your hair, you need to take care of your hair if your hair texture should be good, but that is not always true. Everything depends on the gene. If enough amount of keratin protein gets produced, then only our hair will look lustrous, it will look healthy and all that. Similarly, our nails also, right? So everything depends on the protein that is produced. So likewise, a particular gene on the DNA strand is responsible for the production of a protein, which is nothing but an enzyme, and that enzyme has a specific function. We'll understand that better by taking the example. Say for example, if an enzyme called as beta-galactosidase. So we'll take the example of a bacteria, that is E. coli. So this E. coli bacteria, it produces an enzyme. One of the gene in the E. coli bacteria produces an enzyme beta-galactosidase. So this beta-galactosidase has a particular function, because a particular enzyme will have a particular function. What is the function? We shall look into. Its function is to catalyze or increase the rate of hydrolysis of a disaccharide molecule. What disaccharide molecule? It will help in, or it will increase the rate of hydrolysis of lactose into galactose and glucose. So what did we understand now? If in our body lactose, we taken lactose, right? That lactose, if it has to get converted into galactose and glucose, what is needed? Beta-galactosidase is needed. That was studied by taking the example of E. coli. So in E. coli, there was an enzyme which is called as beta-galactosidase. That beta-galactosidase has a particular function wherein it increases the rate of reaction of hydrolysis of lactose into galactose and glucose. Therefore, again I am repeating, what did we understand? If lactose has to be broken down into galactose and glucose, which enzyme is needed here? Beta-galactosidase. Now, this bacteria uses beta-galactosidase as a source of energy, right? Because that energy is the one which breaks the lactose into galactose and glucose. Now, imagine if that gene, because of a mutation, that gene did not produce beta-galactosidase. What will happen? Lactose will not get converted into galactose and glucose. If lactose doesn't get converted into galactose and glucose, it will hinder or alter various metabolic processes in the body. So that is why one particular enzyme is responsible for the functioning of, or for doing a particular mechanism of function in the body of an organism. Hence, if the bacteria, as I told you, if the bacteria do not have lactose around them to be utilized for energy, they would no longer require the synthesis of the enzyme beta-galactosidase. So therefore, it is the metabolic, physiological or the environmental condition that regulates the expression of the gene. So therefore, lactose is the one that provides the energy. It is the energy source that is utilized by the bacteria. And if that lactose has to be utilized as an energy source, beta-galactosidase enzyme is needed, wherein the lactose gets converted into galactose and glucose. So the development and differentiation of, so one more example here, the development and differentiation of embryo into an adult organism. All these also are a regulated, coordinated process which requires the expression of several sets of genes. So therefore, any process, be it metabolical, physical, or any process that is occurring in a body, For that, a particular enzyme will be produced that trigger the or catalyze the various mechanisms which is needed for the particular function to occur in a body. One more example that we had taken is that how an embryo gets converted into an adult. Do you think that only cell division is enough if an embryo has to get converted into an adult? No. How many fingers has to get formed, how many toes has to form, what the skin texture should be, how big the eye should be, how the organ should look like, how the organ should function. Each and every tiny thing in our body is regulated by the genes itself, and each and every function is done by a specific gene, or one gene can perform 2 or 3 functions. So therefore, the expression of genes is very important in any of the organism if the normal individual has to live happily without any deformities. So that is why it is very important. So next, let us look into the regulation of gene expression in the case of prokaryotes. So in prokaryotes, the control of the rate of transcriptional initiation is very important for the control of gene expression, because we all know that there is an initiator region, right, wherein the initiation of transcription begins. And how that initiation happens, how it is controlled, always decides how the genes has to get expressed. And in a transcription unit, the activity of RNA polymerase enzyme. Why is an RNA polymerase enzyme here? Because we are synthesizing, that is during the process of transcription, what gets formed? mRNA strand forms. mRNA strand, if it has to get formed, which is complementary to a DNA strand, then a DNA dependent RNA polymerase enzyme is important. So the activity of the RNA polymerase enzyme at a given promoter region is regulated by the interaction with the accessory protein. So therefore, if RNA polymerase, which is an enzyme, also has to get formed, what will the RNA polymerase do? It will catalyze the reaction wherein it will begin the process of transcription or the formation of the mRNA strand from the initiation site up till the termination site, right? Now, from where will the RNA polymerase get produced? Again, RNA polymerase is an enzyme which is regulated, which is formed by the interaction of various accessory proteins. How are these proteins produced? We had studied earlier, and which affects its ability to recognize the start site. So these regulatory proteins can act both positively. So these regulatory proteins can regulate or activate the process of transcription, or activate the process of formation of a mRNA strand, or they can stop the process of formation of mRNA strand. So what these proteins will do is they will go and bind to the promoter region. After binding, either they can activate transcription. Activating the transcription means they can trigger the RNA polymerase to produce the mRNA strand, or they can make the RNA polymerase to remain silent and not allow it to produce the mRNA strand. So two things can happen. So therefore, the regulator proteins can act positively, wherein they can act as activators, and they can act negatively, wherein they can act as repressors. How? We shall look into by taking an example of lactose in the further slides. Accessibility of promoter region of prokaryotic DNA is many cases regulated by the interaction of proteins with sequences termed as operators. So in the operator region, there are various proteins that actually regulate the expression of genes in the case of the prokaryotes. And the operator region, where is it present? It is present exactly adjacent to, that is near to the promoter region in most of the operons, and in most cases the sequence of operator bind a repressor protein. So here, what goes and binds to the operator region will decide whether the initiation of transcription should occur or it should not occur. So this thing, when I explain it to you taking an example, you will understand it better. So each operon has a specific operator and a specific repressor for it. So if there is a specific operator, it will go and bind and it will promote the transcription process. If a repressor goes and binds, it will stop the transcription process, or it will not allow the transcription to start at all. So if transcription doesn't occur, mRNA will not get produced, and if mRNA doesn't get produced, then translation will not occur. No translation, then no protein synthesis. No protein synthesis, then the individual will be subjected to a lot of disorders. So that is how it occurs. So we shall take the example of regulation of gene expression in prokaryotes by taking the example of a Lac operator concept. This is called as the Lac Operon concept. This Lac Operon concept is one of the important concepts that they ask in the exam. So in prokaryotes, we shall take an example. So here I told you there is a operator region present, there is a promoter region present, right? And there are different operators that are present. So here we will consider about the Lac operator. So the Lac operator is present only in the Lac Operon. Now what is this Lac Operon? Don't think it is something from somewhere it comes. It is nothing but a strand on the DNA. That particular strand on the DNA is called as Lac Operon. Okay. In that Lac Operon, there is one small segment of DNA which is called as the Lac operator, where it is present only in the Lac Operon, and it interacts specifically only with the Lac repressor. Now here I'll show you. This is a Lac Operon. What is this Lac Operon? This Lac Operon is nothing but a segment of the DNA strand. In that segment, what all are present? There is presence of promoter region, there is presence of operator region, and there is presence of different structural genes. Remember, in the previous session, you have studied, right? That is, we had studied about the transcription unit. What was there in the transcription unit? Promoter region was there. We studied about the structural genes. Then we studied about the terminator region, right? But here, after the promoter region, there is one region which is called as the operator region, and there is the structural genes that present. So this particular sequence which is having the operator region, that is the Lac operator region, it is called as the Lac Operon sequence. So they have taken the example of a Lac Operon sequence. No, so now what is needed if the process of transcription has to occur? A inducer is needed, right? What happens if an inducer is not there? We shall look into. So when an inducer is not there, so if an inducer is not there, what happens? Actually, an inducer should go and bind. So before moving to that, we shall study about what all these means. So here there is the presence of a region which is called as I. That is the regulatory gene inhibitor which codes for the repressor of the Lac Operon. So this region codes for the repressor of the Lac Operon. Repressor means that suppresses something, right? So here that is I region, which is the inhibitory region. Next comes the Z, Y and A, which forms the structural genes. So here Z, so this one, Z, Y and A, they form the structural genes, because after the promoter region, structural genes are present on the transcription unit. We had studied about that, right? So Z gene codes for beta-galactosidase. If you remember there, what was beta-galactosidase? Beta-galactosidase is an enzyme that helps in the breaking down of lactose into glucose and galactose. We had studied, right? So beta-galactosidase enzyme is produced by which gene? Z gene. Now here, I'll write it here, beta-gal. It is produced by this particular Z gene. So beta-galactosidase, which is primarily responsible for the hydrolysis of lactose into galactose and glucose. And next, one more gene is there, Y gene. Y gene codes for an enzyme which is called as permease. So what does Y gene code for? It codes for an enzyme which is called as permease. And what is this permease enzyme important for? Permease enzyme is important for permeability of the cell to beta-galactosidase. The cell has to absorb beta-galactosidase. So how much beta-galactosidase has to enter into the cell will be decided by this particular Y gene. Then the A gene that is there, it codes for trans-acetylase. So this codes for trans-acetylase. Now we have understood this is the inducer region, promoter region, operator region. These are the structural genes wherein the Z gene codes for enzyme beta-galactosidase, the Y gene codes for enzyme permease, and the A gene codes for enzyme trans-acetylase. There are various other genes also present, but we are talking about only about the Lac Operon region, right? That is why we are choosing only so much, because only so much is the Lac Operon region. Now, a inducer is required. Why is the inducer is required? If the inducer is required, then only the mRNA strand will get produced. Now, what happens when an inducer is not there? We shall look into. So, in the absence of inducer, the repressor will bind to the operator region. What is a repressor? Repressor means it is the one that represses something, that stops something. Now, if the promoter region has to get activated, then an inducer should be present. Understood? If the inducer is not there, what will happen? In that place, a repressor will go and bind. So, if the repressor will go and bind there, then it will, what is that? It will not allow the production of the RNA polymerase by the promoter region. It will not help in the binding of the RNA polymerase in the promoter. If their RNA polymerase does not bind, then from where will transcription occur, right? And if transcription doesn't occur, then production of lactose will not take place. So therefore, the conversion of lactose, galactose and glucose, all that will not occur. It will stop. So therefore, inducer is very, very important. So that is what they're saying. So the proteins, they can be both activate a transcription process or they can repress the transcription process. So when the inducer is not there, what happens? To that particular place, the repressor will go and bind. The repressor will go and bind in the operator region. So when the repressor goes and binds in the operator region, it will prevent the RNA polymerase from binding to the promoter region. So when RNA polymerase, that also transcription, please go and look into the transcription session so that you will understand it better. RNA polymerase doesn't go and bind to the promoter region. There is no RNA polymerase there. No RNA polymerase means mRNA strand will not get produced, because if mRNA strand has to be produced, then RNA polymerase should be there, right? So that is how it will stop the transcription process, or it will stop the expression of a particular gene. Now on the other hand, we'll take another example. Now this was in the absence of an inducer. Now if an inducer is there, what will happen? We shall look into. In the presence of inducer. In the presence of an inducer, what will happen is, this inducer, it will go and bind to the repressor region. It will go and bind to the repressor region. Therefore, it will not allow the repressor to go and bind to the operator region. So when the repressor doesn't bind to the operator region, RNA polymerase will go and bind here. RNA polymerase will bind. So if RNA polymerase binds, then the mRNA strand will get produced. What mRNA strand? Lac mRNA strand will get produced. So what is this Lac mRNA strand if it gets produced? Then in that Lac mRNA strand, what all will get produced? Beta-galactosidase will get produced, permease will get produced, trans-acetylase will get produced. That is, translation will occur. So transcription and translation both will occur in the presence of inducer. Transcription will not occur in the absence of inducer. So therefore, the translation will also not occur. So I hope you understood. If an inducer is not there, then what will happen? The repressor protein, it will act like a negative. It is negative in nature now. It will go and bind to the operator, therefore preventing the binding of the RNA polymerase to the promoter region. And if RNA polymerase doesn't bind to the promoter region, the mRNA strand will not get produced. If the mRNA strand does not get produced, how will it read the structural gene, right? So therefore, the lac mRNA will not get produced. Therefore, galactose and glucose will not occur. And in the presence of inducer, what will happen? That inducer will go and bind to the repressor. If it goes and binds to the repressor, it will inactivate the repressor. Therefore, the repressor will not go and bind to the operator region. If the repressor doesn't bind to the operator region, then the RNA polymerase can freely go and bind to the promoter region. And when the RNA polymerase freely binds to the promoter region, it will promote the transcription. Means the mRNA strand codes for which is complementary to the structural genes will get formed. Therefore, the lac mRNA strand will get formed, and this lac mRNA strand will get translated. It will enter into the translation process wherein it will produce the beta galactosidase, permease and trans-acetylase. So this is how gene expression occurs in the case of prokaryotes. Therefore, the presence of an inducer and the absence of an inducer plays a very important role here. Inducers and repressors both are present, but so inducers and repressor both are there. So repressors are always present, but whether the repressor will go and bind to the operator region or not depends on whether there is presence of an inducer or whether there is an absence of an inducer. So this is about the Lac Operon concept. So because this kind of gene expression was expressed using the particular sequence which was responsible for producing the lac mRNA, it is called as the Lac Operon concept. So this was about Lac Operon concept. So next, moving on to the Human Genome Project. So Human Genome Project is a very big project that was launched in the year 1990. So what did they actually do in this project? In this project, actually, they sequenced the entire human genome. We all have a stretch of DNA, right? Our cells, how many cells our body has, in so many cells also the DNA strands are present. So the entire DNA strand was sequenced, but the beginning of sequencing started in the year 1990. And for that, they opened a particular project which was called as the Human Genome Project. So the Human Genome Project, it is a 13-year project and it was coordinated by the United States Department of Energy as well as the National Institute of Health. So both the United States Department of Energy and the National Institute of Health, they came together, put forward an idea of sequencing the human genome so that various diseases, disorders, everything can be studied easily by using the computers itself. Sitting where you are, you can just using the computer, looking into the sequence, you can do various kinds of experiments. So during the early years of the Human Genome Project, the Wellcome Trust, which was there in United Kingdom, it became a major partner, and after that other contributors also came forward from Japan, France, Germany, China, and the project was completed in 2003. So therefore, it was a collaborative work among different institutes of different countries. And human genome, as we know, after that, after the Human Genome Project, after complete sequencing of the human genome, we came to know that the human genome is said to have approximately 3 into 10 to the power 9 base pair. So after the complete sequence, it was then we came to know that the length is about 3 into 10 to the power 9 base pair, and the cost of the entire sequencing was about $3 per base pair. That is, the estimated cost in the beginning was $3 per base pair. To sequence it, to do various experiments, sequence it, it cost about $3. And the total estimated cost of the project at the end would be approximately $9 billion. So imagine how big this Human Genome Project was. But though so much money had to be spent, it was of great importance in the medical field especially. And further, if the obtained sequences were to be stored in typed form in the form of books, and if each page of the book contained 1,000 letters and each book contained 1,000 pages, then 3,300 such books would be required to store the information, right? Because imagine that long stretch of DNA, entire sequence, if it has to be stored, then 3,300 books are needed. So that is why it is not stored in the typed form, but it is stored in certain information centers, and that information centers is called as bioinformatics. So what is bioinformatics? So bioinformatics is nothing wherein it's like a program. So a computer will be there. In that computer, the programs will be done, and in that programs or in that app itself, the information of the DNA sequences will be stored, so that it need not be printed in a book. Because as I told you, if it has to be that entire length of sequence of 3 into 10 to the power 9 base pair length of human genome, if it has to be sequenced and if it has to be typed in the book, then it will require 3,300 books. So to avoid that, the bioinformatics came into existence, and bioinformatics is now today a very vast subject and it has a good scope. So whoever wants to take up bioinformatics as their higher studies, it is one of the best options to choose, because it has a lot of scope, because very few people go into bioinformatics. So we'll study about bioinformatics, what it is exactly. So the enormous amount of data that is there. So instead of typing, I told you, if it was supposed to be typed, 3,300 books are needed to store the sequence, to type the sequence of one DNA strand, right? So instead of doing that laborious work, the enormous amount of data expected to be generated also necessitated the use of a high-speed computational device for data storage and retrieval. Now also we have certain websites. Under bioinformatics, there are certain websites called as NCBI and all that, wherein just sitting in your home, in your fingertips, you log into NCBI, and whatever sequence of any organism you want DNA sequence, you'll get it in that website. It's very easy, in your fingertips only you'll get all the information. So Human Genome Project was closely associated with the rapid development of a new area in biology, which was called as bioinformatics. Bio is something related to life. So all the informations related to life, starting from the DNA or information about each gene, what do they produce, what type of enzyme they produce, what type of proteins they produce, everything is stored in the computer in the form of a app. That information is stored in the computer. That is nothing but bioinformatics. So some of the goals of HGP we shall look into. So some of the important goals of Human Genome Project are to identify all the approximately 20,000 to 25,000 genes in the human DNA. So imagine 20,000 to 25,000 genes are there in the human DNA. Those genes will code for specific amino acids, therefore they produce specific proteins or enzymes, and there are some genes which can produce 2 or 3 such proteins or enzymes, right? So all that information should be finished with a certain span of time, should be collected, the data stored in bioinformatics. So that was their goal. And to determine the sequence of the 3 billion chemical base pairs that make up the human DNA. Imagine how much the human DNA is made up of 3 billion chemical base pairs, and cracking those 3 billion chemical base pairs and storing them as a data in bioinformatics, that was also one of their goals, and to store information in the form of databases. Now there are a lot of websites that are available which stores information in the form of databases, and also to improve tools for data analysis. So how to interpret that data, for that also there are various tools that are pre-programmed in the computer or apps developed for that. So of using all those genetic tools also to improve those tools, that was also their aim. And transfer related technologies to other sectors such as industries, and to address the ethical, legal and social issues that may arise from the project. So all these were the goals. So ethical issues, so all these can lead to the curing of a disease and all that, but should they opt for that or not? What if they opt for that? Because opting for that might make human beings immortal, which is not good for the world, right? Which is not good for the balanced ecosystem. So what might happen if they do all that? What ethical, legal and social issues might rise? So all that they had to keep in mind and frame certain rules and regulations according to that. So all these were the goals of the Human Genome Project. Now let's look into the importance of Human Genome Project. So the knowledge about the effects of DNA variations among individuals. So we all know that each individual has their own set of DNAs, and these DNA variations, what are the effects of these DNA variation, whether these DNA variations can lead to can be used as a diagnosis tool, and whether it can be used to treat people and cure them of diseases and prevent thousands of disorders that affect the human beings. All these can be done by Human Genome Project, because if you need to know understand a genetic disorder, just sit at your home, click on that particular sequence, why that particular sequence is, or gene is causing this particular genetic disorder. You'll get all details about it. So that is the importance of HGP. And apart from providing clues to understand human biology, they also help in learning about the non-human organisms' DNA sequences. So, the other organisms' DNA sequences. Easy example, I'll just take a monkey or a rat. So, whenever any study has to be conducted on human beings, rat is used as a laboratory tool, right? It is said that the gene of rats and human beings match to a greater extent. So how did scientists come to know that? It is because of sequencing. So that is why, apart from giving importance to sequencing only the human genes, even the sequencing of the genes of other organisms are also given importance in the Human Genome Project. All these sequencing can actually be applied to solving challenges in healthcare, to overcome agricultural problems, in energy production, environmental remediation. For all these it can be used. Say for example, environmental remediation. A particular gene in a particular bacteria can be altered so that that bacteria can be used for the cleaning of the environment. So like that, for all that, if the data is available in the computer, you can just do it there itself and then go in for laboratory study. So that is the importance of HGP here. And what are the various methodologies that are adopted in the Human Genome Project? So for sequencing and for all that, what are the different methodologies they had adopted? We shall look into. So first methodology they had used was expressed sequence tags. So express sequence tags, they are mainly focused on identifying all the genes that are expressed on the RNA. Because how did they know that each gene produces a particular type of protein or an enzyme? They had to sequence it, they had to find out, right? So that was done using express sequence tag. So this is one of the tool. And next is by sequence annotation. So what they do in sequence annotation is simply they'll sequence the whole set of the DNA genome, and that DNA genome will contain both the coding and the non-coding sequences. That is the exons and the introns, both will be there, and later they will assign the different regions, what different functions they perform, all that will be assigned. So that is about sequence annotations. So sequence annotation was one of the important tool that was used. Next, how did they do the sequencing for HGP? We learned that they did the sequencing of 3 into 10 to the power 9 base pair length of DNA. And how exactly was the sequencing done? What methodology was followed? We shall look into. For sequencing the total DNA from a cell of an isolate cell, so how exactly the sequencing has to be done? So for sequencing, first a DNA is required, right? That DNA strand has to be isolated from the cell. Why from the cell? Because the cell will have nucleus. Inside the nucleus, chromosome is there. What is that chromosome? Nothing but it contains DNA strand. So if we need to isolate the DNA strand, first we need to separate the cell. So first the total DNA from the cell is isolated. Then total DNA is isolated. Then that DNA will be converted into random fragments of relatively smaller sizes. So at a stretch, you cannot sequence that 3 into 10 to the power 9 base pair of DNA. It's difficult. So that is why they break it into short fragments. And after breaking into short fragments, then they are cloned. So one small stretch of DNA is not sufficient for the experiment, because the experiment has to be repeated in trials. If you need to repeat the experiment in trials, you need more number of the same clone of that DNA strand, right? So for that, they will clone this DNA strand by using cloned in suitable host using specialized vectors. They will clone it using specialized vectors. And how is the vector? So vectors can be either a bacteria. Say for example, one best example for a vector is E. coli. E. coli is a vector. All that you will learn in the coming chapters, and you'll understand that very, very clearly. So E. coli, that is in biotechnology applications, you'll study there. So it will be cloned in vectors. So what they'll do is, they'll isolate they will take a cell. From that cell, they will isolate the DNA. That DNA is very, very lengthy. So entire sequencing is difficult at a time. So what they'll do is, they'll break the DNA strands into smaller pieces of fragments. And those fragments, they will clone it. They will make multiple copies of those fragments. Copies means, you know, right? They look exactly like the parental DNA strand that was taken. So that clone, now for cloning, how do they clone it? What they'll do is, they'll take that particular DNA strand and put it into a vector, say for example, into a E. coli. Now when that E. coli bacteria multiplies, that DNA strand also will multiply in the bacterial cell, right? Therefore, the number of clones of the DNA fragments will be got. So that is how they will do it. Now the cloning resulted into amplification of each piece of DNA. That is, each piece of DNA, one DNA was isolated. It was broken into 10 fragments. Now for that, each 10 fragments, 10, for example, fragment 1, for that same copy, 10 copies will get produced. Now that is called as amplification. Amplification means increasing in the number of the copies of the DNA strand. That will be amplified, and that will be subsequently sequenced very easily. For sequencing, what they will do, they will put it into an instrument which is called a sequence analyzer. That sequence analyzer will give data in the computer. This is the sequence. So can you see here on the computer, the sequence got seen. So every fragment sequence they will get, and then they will join the sequence and make it into a stretch. Now we'll understand, in that stretch of DNA, that is 3 into 10 to the power 9 base pair of DNA, these are the sequences that are present. So that is how sequencing is done in Human Genome Project. And the commonly used host were bacteria and yeast, as I told you. They are put into a vector, right? So usually bacteria and yeast are used, and the vectors were called as BAC for bacterial artificial chromosomes. If bacteria is used as a vector, that vector was called as BAC, which means bacterial artificial chromosome. And if a yeast is used as a vector, it is called as YAC, YAC, that is yeast artificial chromosomes, because their artificial chromosomes are produced, their clones are produced, right? So that is why. And the fragments are sequenced using automated DNA sequences that worked on the principle of method developed by Frederick Sanger. So Frederick Sanger was the first person to develop an automated sequencing machine which will display the sequence of that particular strand of DNA on the computer screen. So these sequences were then arranged based on some overlapping regions present in them. So they are arranged. They have to be arranged, right? Why? Because small fragments are sequenced. At a stretch, the DNA was not sequenced. So that is why. And this required the generation of overlapping fragments for sequencing, and alignment of these sequencing was very difficult. It was not that possible. Therefore, later on, specialized computer-based programs were developed. And these computer-based programs are developed in such a way that the computer itself will arrange the sequences and give you, at a stretch, it will give you the complete sequence of the DNA strand. So these sequences were subsequently annotated and were assigned to each of the chromosome. So the sequence of chromosome 1, so DNA, how many chromosomes? Say for example, we are talking about Human Genome Project, right? So therefore humans have how many chromosomes? 46, right? 23 pairs of chromosome. In that, the first chromosome that is there, right? In that first chromosome, DNA was there. That DNA, what sequence is present, that was identified in the year, that complete sequencing was completed in the year May 2006. Think of it, Human Genome Project started in 1990, but the first chromosome was completely sequenced, or the DNA present in the chromosome 1 of the human DNA was completely sequenced in May 2006. How many years they took, what lot of laborious work, right? So this was the last of the 24 human chromosomes, wherein 22 autosomes X and Y to be sequenced. So therefore, later on, all the chromosomes of the DNA, including the autosomes, that is the female chromosome, X and Y chromosome, were all sequenced, and the complete sequencing was done and it was completed. So another challenging task was assigning the genetic and physical maps of the genome. So that because a lot of genes are there, and those genes, it was very difficult to analyze a genetic and a physical map for them, and this was generated using information on polymorphism of restriction endonuclease recognition sites and also some repetitive DNA sequences. Why was it difficult? Was because some of the DNA sequences will get repeated. So understand that and all that, it was very much difficult. And these repetitive DNA sequences are known as microsatellites, which is very, very important in DNA fingerprinting. So that is why, because DNA is unique in each of the organism or in each of the human being itself, for DNA fingerprinting and all that, it can be used. That is the repetitive sequences, which are called as the microsatellites. So let us study about the salient features of the Human Genome Project. So the human genome, what did they find out? They found out that the human genome contains 3164.7 million base pairs. It is made up of that. Imagine sequencing such a lot of lengthy human genome, how difficult it might have been. And the average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin. So which is the largest human gene that is present? It is called as dystrophin, and it has and the sequence length of that dystrophin is 2.4 million bases. It is made up of 2.4 million bases. It is the largest gene that is produced by the human DNA, and the total number of genes is estimated at 30,000, much lower than the previous estimate of 80,000 to 1,40,000 genes. And almost all 99.9% of nucleotide bases are exactly the same in almost all the people. So nucleotide bases are the same, but in spite of that, they are, because of the microsatellites present and because of all that different, that is because of some light variation through DNA fingerprinting, various things can be done. So this is about the salient features. It was unknown for about over 50% of the discovered genes. So so many genes were discovered by the Human Genome Project. 50% of the genes function was still unknown, and less than 2% of the genome codes for proteins. That is what they found out. And the repeated sequences make up a very large portion of the human genome. And because of this, it was quite a laborious process. And repetitive sequences are stretches of DNA sequences that are repeated many times. Sometimes it is repeated hundreds to thousand times, and therefore they are said to have no particular coding function, because the same sequence gets repeated thousands to 10,000 times, but they shed light on chromosome structure, dynamics, and evolution of the organism. So it is said that as and when the organism evolves, the number of repetitive sequences in that particular organism tends to increase. Maybe that provided a clue for the evolution and evolutionary relationship of an organism or everything. So these were the salient features of the Human Genome Project. Apart from that, they found out that chromosome 1 is the one that has the major number of genes, that is about 2,968 genes are present in chromosome 1, and that is one more gene, the chromosome Y gene, which is present in female. It has the most lesser number of genes, that is only 231 genes are present in chromosome Y. So the X chromosome of chromosome 1 has 2968 gene, whereas the Y chromosome, which is present in females, has only 231 genes. And scientists, they have also identified about 1.4 million locations where single base DNA differences, which are called as single nucleotide polymorphism, or it is also called as SNPs, occur in humans. And this information promises to, because of this single basis DNA differences, it is possible for us to trace the human history and all that. And not just that, it is possible for people to identify a culprit through that is, if forensic department, for them to identify the fingerprinting, the DNA sample and all that is possible because of the presence of the SNPs in the DNA. So that is when DNA fingerprinting and all that comes to help. So this was about the salient features of the DNA. So I hope you understood the session well. In the coming session, let us talk about a very interesting topic, that is DNA fingerprinting, and let's understand what are the techniques that are followed in DNA fingerprinting. It is a very interesting topic. So we shall meet again in the coming session and discuss about this particular topic. Thank you.